Retrovirus nature. Retroviruses, an intriguing class of RNA viruses. Retroviruses belong to a unique family of RNA viruses distinguished by their remarkable ability to replicate their genetic material in a reverse manner. The term retro refers to this distinctive reverse transcription process, a fundamental characteristic that sets retroviruses apart from other viruses. Unlike most viruses that utilize DNA as their genetic material, retroviruses carry their genetic information in the form of RNA. Reverse transcription, unraveling the genetic puzzle. The hallmark of retroviruses is their use of an enzyme called reverse transcriptase. Upon infecting a host cell, retroviruses, including HIV, reverse transcribe their single-stranded RNA genome into double-stranded DNA. This process is akin to turning the usual flow of genetic information on its head, as genetic material is typically transcribed from DNA to RNA during gene expression. The reverse transcription process involves several crucial steps. First, the retrovirus enters the host cell and releases its RNA genome. Reverse transcriptase then catalyzes the synthesis of a complementary DNA strand, producing a hybrid molecule known as RNA-DNA duplex. This duplex subsequently undergoes further enzymatic processes, resulting in the generation of a complete double-stranded DNA molecule. This viral DNA is then integrated into the host cell's genome, becoming a permanent part of the cellular DNA. Integration into the host genome, a viral residence. One of the key features of retroviruses is their ability to integrate their genetic material into the host cell's DNA. This integration is facilitated by another viral enzyme, integrase. The integrated viral DNA, now termed provirus, essentially becomes a part of the host genome. This integration allows the virus to persist within the host cell for an extended period, remaining latent or actively producing new virus particles. The provirus may lay dormant within the host cell, escaping the immune system's detection. Alternatively, it may become transcriptionally active, leading to the synthesis of new viral RNA and proteins, ultimately resulting in the assembly and release of new virus particles. The integrated nature of the provirus poses challenges for the immune system and complicates efforts to eradicate the virus entirely, making retroviruses a particularly elusive class of pathogens. Latency and the challenge of eradication. The ability of retroviruses to establish latency adds an additional layer of complexity to understanding and treating infections. Latency occurs when the integrated provirus remains dormant within the host cell, evading the immune system's surveillance. This latent phase can persist for an extended period, and the virus may later become reactivated, leading to renewed viral replication and potential progression of the infection. The concept of latency is particularly pertinent to HIV, the human immunodeficiency virus. In HIV infections, certain cells, such as long-lived memory CD4+ T cells, can harbor latent proviruses. These cells serve as a reservoir from which the virus can re-emerge, even after prolonged periods of antiretroviral therapy. Consequently, eradicating HIV from an infected individual poses a significant challenge due to the potential for viral reactivation from these latent reservoirs. Evolutionary adaptation and genetic diversity. The retrovirus nature of HIV contributes to its high genetic variability. Reverse transcription, a process prone to errors, leads to the accumulation of mutations in the viral genome. Additionally, the activity of reverse transcriptase lacks the proofreading mechanisms present in cellular DNA replication, further increasing the mutation rate. The consequence of this high mutation rate is the existence of diverse viral strains and subtypes. These variants may differ in their biological properties, including transmissibility, pathogenicity, and resistance to antiretroviral drugs. The continual evolution of HIV presents a formidable challenge for the development of effective vaccines and treatments, as the virus can adapt rapidly to selective pressures. The retrovirus nature of HIV underlies its distinctive life cycle, including reverse transcription, integration into the host genome, and the establishment of latency. These features contribute to the challenges in treating and eradicating HIV, emphasizing the need for ongoing research to unravel the complexities of retroviral infections. Targeting the immune system, HIV's strategic assault. HIV, the human immunodeficiency virus, is characterized by its strategic and specific targeting of the immune system, the body's primary defense mechanism against infections. This unique aspect of HIV's behavior is a key factor contributing to the progressive weakening of the immune system, ultimately leading to the development of acquired immunodeficiency syndrome, AIDS. CD4 plus T cells, ground zero for HIV. At the forefront of HIV's assault are CD4 plus T cells, a subset of white blood cells that play a central role in orchestrating the immune response. These cells are equipped with the CD4 receptor, which serves as the primary docking site for HIV. The virus gains entry into the host cell by binding to the CD4 receptor, initiating a series of events that allow it to infiltrate and hijack the cellular machinery. Once inside the CD4 plus T cell, HIV employs its reverse transcriptase enzyme to convert its RNA genome into DNA. This newly formed viral DNA is then integrated into the host cell's genome, turning the infected cell into a factory for producing more virus particles. The relentless replication of HIV within CD4 plus T cells leads to their depletion, directly compromising the immune system's ability to mount an effective defense against infections. Gradual erosion of immune function. As the infection progresses, the continual destruction of CD4 plus T cells weakens the immune system, leaving the body vulnerable to opportunistic infections and certain cancers. The hallmark of HIV infection is the progressive decline in the number of these crucial immune cells. The virus's preference for CD4 plus T cells, which are central to coordinating immune responses, contributes to the intricate and strategic nature of HIV's assault on the immune system. Implications for immune response. HIV's targeting of the immune system has broader implications beyond the direct depletion of CD4 plus T cells. The virus also disrupts the delicate balance of the immune system by impairing the function of other immune cells and components. For example, HIV can interfere with the activity of dendritic cells, which are essential for initiating immune responses, and impair the function of cytotoxic T cells, which normally play a crucial role in eliminating infected cells. The chronic inflammation induced by ongoing viral replication further exacerbates immune dysfunction. Elevated levels of immune activation contribute to the exhaustion and dysfunction of immune cells, creating a microenvironment that is conducive to viral persistence and disease progression. Clinical manifestations from HIV to AIDS. The consequences of HIV's targeted assault on the immune system become clinically apparent as the infection advances to AIDS. With a severely compromised immune system, individuals with AIDS are susceptible to a range of opportunistic infections and cancers that would not normally pose a threat to those with a healthy immune system. Pneumocystis pneumonia, Kaposi's sarcoma, and tuberculosis are examples of conditions that can manifest in the later stages of HIV infection. HIV's ability to specifically target and deplete CD4 plus T cells, along with its broader impact on immune function, distinguishes it as a unique pathogen. Understanding the intricacies of this targeted assault is crucial for developing therapeutic strategies to mitigate the effects of HIV on the immune system and improve the prognosis for individuals living with HIV/AIDS. High genetic diversity in HIV, the shifting landscape of an adaptable virus. IV, the human immunodeficiency virus, stands out for its remarkable genetic diversity, a characteristic that poses significant challenges in the realms of treatment, prevention, and vaccine development. This diversity is a consequence of the virus's high mutation rate and the existence of various subtypes and recombinant forms, creating a dynamic and adaptable viral landscape. Mutation rate and genetic plasticity. HIV's genetic diversity is intricately linked to its inherent high mutation rate during the replication process. Unlike many DNA-based organisms, the reverse transcriptase enzyme used by HIV lacks proofreading mechanisms, leading to a higher likelihood of errors during the synthesis of viral DNA. This increased error rate contributes to the rapid accumulation of mutations in the viral genome, fostering genetic diversity within and between infected individuals. The plasticity of the virus extends beyond simple point mutations. HIV is prone to genetic recombination, a process where two different viral strains co-infect a single host cell, leading to the exchange of genetic material. This phenomenon results in the creation of novel recombinant forms, adding another layer of diversity to the viral population. Subtypes and recombination, an evolving mosaic. HIV is categorized into several subtypes, A, B, C, D, etc., each with distinct genetic characteristics. Subtypes are geographically distributed, contributing to the global mosaic of HIV diversity. Moreover, the virus often undergoes recombination, giving rise to circulating recombinant forms, CRFs, that carry genetic material from different subtypes. This dynamic process continually generates new variants of the virus, each with its own unique genetic makeup. The implications of HIV's genetic diversity extend to various facets of the epidemic. For instance, different subtypes may exhibit differences in transmissibility, pathogenicity, and response to antiretroviral therapy. The emergence of drug-resistant strains is also influenced by the genetic diversity of the virus, making treatment regimens more challenging to formulate and implement effectively. Challenges for treatment and vaccine development. The high genetic diversity of HIV poses significant obstacles for the development of effective treatments and vaccines. Antiretroviral drugs, which have revolutionized the management of HIV, often target specific viral proteins. However, the diversity of these proteins among different viral strains can result in variations in drug efficacy and resistance patterns. Developing a universally effective HIV vaccine is particularly challenging due to the virus's ability to mutate and evade immune responses. The immune system, which typically relies on recognizing specific viral proteins, may struggle to mount an effective defense against the diverse array of HIV variants. The continuous evolution of the virus necessitates ongoing research efforts to understand and address the intricate interplay between HIV's genetic diversity and the host immune response. HIV's high genetic diversity is a defining feature that shapes the course of the epidemic and complicates efforts to control and eliminate the virus. The adaptability conferred by this diversity underscores the need for comprehensive and innovative approaches in the ongoing quest to combat HIV/AIDS. Latency and integration into the host genome, HIV's stealthy persistence. One of the distinctive features of HIV, the human immunodeficiency virus, lies in its ability to establish a persistent infection through latency and integration into the host genome. This complex interplay between the virus and the host cell contributes to the challenges in eradicating HIV and understanding its long-term impact on infected individuals. Latency, the silent phase of infection. Following the initial infection of a host cell, HIV has the capacity to enter a state of latency. During this phase, the virus becomes quiescent, ceasing active replication and evading detection by the immune system. Latently infected cells act as reservoirs, harboring the integrated viral genome without producing new virus particles. This ability to remain silent within the host cell allows HIV to evade antiretroviral treatments and persist over extended periods, even in the presence of therapeutic interventions. The establishment of latency is particularly evident in long-lived immune cells, such as memory CD4+ T cells. These cells play a crucial role in maintaining immunological memory, allowing the immune system to respond more rapidly upon re-exposure to a previously encountered pathogen. However, when infected with HIV, these memory cells can serve as a reservoir for latent virus, creating a persistent source of viral reactivation even after prolonged periods of effective antiretroviral therapy. Integration into the host genome, a molecular alliance. Central to HIV's ability to establish latency is its integration into the host cell's genome. This process involves the viral DNA, produced through reverse transcription, becoming a permanent part of the cellular DNA. The enzyme responsible for this integration is integrase, which facilitates the splicing of viral DNA into the host cell's genetic material. Once integrated, the viral DNA lies dormant alongside the host cell's genes. This integration allows the virus to persist and replicate along with the host cell as it undergoes normal cellular processes. The integrated viral DNA may remain dormant for extended periods, evading the immune system's surveillance and making it difficult to completely eliminate the virus from the host organism. Reactivation, the ongoing challenge. The latent phase of HIV infection can be disrupted by various factors, leading to the reactivation of the virus and the resumption of active replication. External stimuli, such as infections or inflammatory signals, can trigger the activation of latently infected cells. Upon reactivation, the integrated viral DNA is transcribed into RNA, initiating the production of new virus particles and the potential for further infection of neighboring cells. The challenge posed by latency and integration is evident in the difficulty of achieving a cure for HIV. While antiretroviral therapy effectively suppresses active viral replication, it does not eliminate latent reservoirs. This latent pool of virus can persist for years, necessitating lifelong treatment to manage the infection. The interplay between latency and integration into the host genome represents a critical aspect of HIV's persistence and the challenges faced in developing a cure. Understanding the molecular mechanisms underlying these processes is crucial for devising strategies to target latent reservoirs and move closer to the goal of eradicating HIV from infected individuals. Transmission routes and stigma. Interconnected challenges in the HIV epidemic. HIV, the human immunodeficiency virus, is not only defined by its biomedical characteristics, but also by the intricate web of transmission routes and the pervasive stigma associated with the infection. The diverse modes of transmission and the societal response to HIV have profound implications for prevention, public health, and the well-being of individuals living with the virus. Transmission routes. Beyond the biological landscape, HIV can be transmitted through various biological fluids, with the most common routes being unprotected sexual intercourse, sharing of contaminated needles, and from an infected mother to her child during childbirth or breastfeeding. The virus is not casually transmitted through everyday contact, emphasizing the importance of understanding and addressing specific risk factors. Sexual transmission remains the primary mode of HIV spread globally. Unprotected intercourse, particularly involving anal or vaginal sex, presents a significant risk, underscoring the importance of safer sex practices and the use of barrier methods, such as condoms. Injecting drug use, sharing needles, and other drug paraphernalia contribute to HIV transmission among people who use drugs. Vertical transmission from mother to child can occur during childbirth or breastfeeding. However, with advancements in medical interventions and prevention programs, the risk of vertical transmission has been significantly reduced, emphasizing the importance of timely and accessible healthcare for pregnant individuals living with HIV. Social stigma, a persistent barrier. In addition to its biological challenges, the HIV epidemic is marked by the social stigma attached to the virus. Stigma refers to negative attitudes, beliefs, and discrimination directed at individuals or groups based on their perceived association with HIV. This stigma is rooted in fear, misinformation, and societal prejudices, creating barriers to effective prevention, care, and support. Stigmatization of individuals living with HIV can lead to social isolation, discrimination in healthcare settings, and reluctance to disclose one's HIV status. Fear of stigma may deter individuals from seeking testing, treatment, and support, hindering efforts to control the spread of the virus and improve the health outcomes of those affected. Intersectionality and vulnerability. Stigma associated with HIV often intersects with other forms of discrimination, such as sexism, homophobia, and racism. Certain populations, including men who have sex with men, sex workers, transgender individuals, and people who inject drugs, are disproportionately affected by both HIV and stigma. The intersectionality of these factors creates additional challenges in addressing the diverse needs of those at higher risk and highlights the importance of comprehensive and inclusive approaches in the HIV response. Educational and advocacy initiatives. Mitigating the impact of stigma requires multifaceted strategies, including education, advocacy, and fostering understanding. Public health campaigns that emphasize accurate information about HIV transmission, prevention, and treatment can contribute to dispelling myths and reducing stigma. Promoting empathy, compassion, and the recognition of the human rights of individuals living with HIV are essential components of efforts to combat stigma. Global impact and future directions. The interconnected challenges of transmission routes and stigma necessitate a holistic and community-driven approach to the HIV epidemic. Addressing the societal factors that contribute to stigma, alongside biomedical interventions, is crucial for creating an environment that supports individuals affected by HIV and encourages open dialogue. As the global community continues to work towards ending the HIV epidemic, acknowledging and addressing the social dimensions of HIV transmission and stigma is integral to achieving meaningful progress.